This dedicated session is for our course orientation. Our subject is S-Biol 418LA. This is Cell and Molecular Biology Laboratory. The section is MEB 34, and I am Arizaldo Castro. I will be your course instructor for the semester. We meet every Mondays and Thursdays, 10:30 to 1:30 p.m. So, a few things for this course orientation. We'll talk about the background of the course, the requirements that we will be asking from you, um, how we do the grading, outline of topics, your expectations. Then we'll also talk about course policies, and in the end, other implementation or other implementing guidelines that we are going to observe for the conduct of this subject for the entire SEM. So let's begin with a few things about the subject. The course is designed for students to be introduced to topics that are uh, laboratory methods and techniques that we use whenever we study cell structure and function. In addition, we are fusing the subject with molecular biotechniques. So at the end of actually at the final modules, the techniques, the methods will be more on the molecular bio part. Some of the methods that we will cover in this subject include uh, the proper usage of CMB instruments in the lab, CMB equipment. Also, we will cover reagent preparation, which is very important, especially if we are actually doing face-to-face setting or we are in a face-to-face setting. You will appreciate how important it is to have a working knowledge on how reagents are prepared, how you should actually prepare certain reagents, no, for each activity. Then we'll also cover techniques and methods on biochemical analysis because we're going to deal with biomolecules. No? We're going to deal with um, proteins, nucleic acids, and carbohydrates and lipids. So definitely, um, how we extract them, quantify them, are key themes in this subject. And lastly, since we're also producing, or we're also going to part of the laboratory for CMB is data analysis and presentation. An important component of the subject is you will learn some computer softwares in order for you to analyze data, to visualize them, to statistically treat them. No? So we designed the subject in a way with one goal, no? Which is for us to promote a deeper understanding of cell and molecular biology to our undergraduate students, especially following the approach of experimentation and research. No? So, since we're doing the subject in a distance learning setup, we have a special provision on the modality and the pedagogy. So that means everything that we're going to or every interaction that we are going to have will be carried out in our digital platforms, in our web available platforms via Schoolbook, MS Teams, and also other communication or other messenger apps. No? So every interaction between you and me will be done through a virtual setting. And all your outputs will be submitted via the Schoolbook e-class that I hope that most of you have already transitioned from the CILP e-class to the official e-class that I created. So all outputs and content will be available via Schoolbook. Okay. Even though we are in the middle of a pandemic, we made sure that the subject is designed to or is compliant with with two important frameworks for higher education, which are the outcome-based approach and the 21st CLD frameworks. So we made sure that the delivery of the topics themselves, are going to be compliant with these two frameworks, especially that we are still in these challenging times that we cannot conduct face-to-face classes. Now, little information about myself as your instructor. I'm also a graduate of our university during my bachelor's, my baccalaureate, and my master's degree. And currently, I'm finishing my Doctor of Philosophy in Biology, major in microbiology at the University of the Philippines. Uh, I have a few institutions. I have worked with a few institutions aside from DLSUD. Right now, I'm also a senior graduate student at our laboratory in UP Diliman at the Microbial Ecology of Terrestrial and Aquatic Systems Lab. I'm a senior graduate student there. I handle data analysis and manuscript writing for one research project of my advisor. And then I also used to teach at LaSalle, DASMAT, before last year. So I actually taught after I graduated. So for 3 years, I taught lecture and laboratory courses in microbiology and cell and mold bio in particular. Um, I also have a working experience with the Department of Health as a research fellow for 2 years. Then afterwards, I returned to teaching again since last year, since quarter 1 of 2020. My research interests as an academic include microbial ecology, especially of aquatic systems, of water systems, natural and built environments. I'm very much interested with elucidating the phenomenon of drug resistance, of antimicrobial resistance in natural aquatic environments as well as built environments. Uh, with my training in bioinformatics, systematics, and phylogenetics, I'm able to have some working projects on these topics. And to a higher extent or to a more generalized extent, I specialize in systems biology, especially animal models. That's why I can actually teach cell and mold bio, microbio, but in the context of relating it to animal and human health. Now, let's move into the requirements. First is the course grading requirement. We follow the absolute grading system, meaning there's no transmutation of your scores, where I'm going to employ the absolute grading system. So whatever you get is going to be computed as is. So for example, you scored 5 out of 10 in a quiz, so that means you get 50% for that quiz. Okay. Last semester, or for the previous semesters that we've been doing distance learning, we followed the grading system where your semestrial grade is the average of your midterm and finals. And for each term, you are given 60% for your enabling assessments and 40% for your summative assessments. As per the newest IRR released by our Vice Chancellor for Academics and Research, there are some revisions to the grading system where we have introduced the component of class participation as 15%. Your enabling assessments are now going to have a lesser value, that's 45%, and your summative assessments will remain at 40%. Your class participation includes your attendance to synchronous classes, meaning we are going, in order for you to get the 15%, you have to be able to attend all the synchronous classes. No? Kasi we've been observing that or I think it's a common observation, or it's a general observation among faculty from different colleges, that since attendance is not is or was not considered for the computation of the grades during the last semesters, there were a few there were students who don't attend the synchronous sessions anymore. So based on the um, based on the discussions from faculty, from the administrators, and also the members of other members of the academic community, it has been resolved that we will include class participation. No? So for my case, I am not only going to include, or I'm not going to solely base the class participation component to synchronous classes. I will also include here participation during discussions, other class participation outputs that I may deem or that I may find necessary during the execution of a particular module. So it's not going to be based solely on your attendance. So class participation, attendance will cover a part of the class participation, but not totally it. So it will still be your choice. For me, I won't be requiring you to attend the synchronous sessions, because I know that there will be, there are differences in terms of your access, your internet connectivity as students from different that are staying in your respective hometowns or provinces. So I understand that. I won't count it personally against you, but it will be counted as class participation. So what I can do, what I can offer is I will add additional components that are not synchronous in order for you to not really have that big of a effect on your class participation if you won't totally be able to attend the synchronous meetings. Okay. Next, the course requirements, the laboratory requirements to be specific. So there will be individual and group requirements depending on what the faculty will be deciding, or what the faculty will, or how the faculty teaching the same subject, how will we decide on the no division of your laboratory requirements, or individual and group requirements. So for group requirements, it may be a worksheet that will be answered as a group, or a presentation that will be given as a group, things like that. For individual naman, we have formative assessments. I am fan of giving formative assessments because even though they are ungraded, these assessments help you prepare for each module. So some of the formative assessments that you can expect to receive from me include reading assignments. I'll provide you some book chapters, journal articles, or online web pages, wiki pages that I want you to read, that I will ask you to read before the module. I may also ask a question following the forum format, and then you participate in the discussion. You get that can be considered as a formative assessment, or it may also be considered as a class participation, component. And then another example, I can ask you to screen or watch a video or a film that is relevant to the discussion. So those will be included, as most of the time such examples are formative assessments. For enabling assessments, which are graded, this include the quizzes that I will give every module, the laboratory worksheets that I will also give every module, and some term or discussion papers which are not necessarily every module, because we have a maximum number of enabling assessments that we can provide to students. So, but at the least, I think I will be providing 2 enabling assessments per module, 1 quiz and 1 laboratory worksheet. For summative assessments, you're familiar with this, but just to reiterate, these are our graded exams, graded summary exams, which we will conduct normally every 2 or 3 modules. For our lab, we are going to have 4 summative exams all throughout the semester. Okay? And we have schedules for that. We have implementing guidelines how we do that. But before I before we have the summative assessment, I ask my students about parameters, or I explain to my students the parameters that I am going to use for the summative assessments. And I ask for their consensus or their suggestion kung okay lang ba sa kanila yung time, okay sa kanila yung what do you call this, yung parameters and all other things that are considered for the execution of the exam. Okay, so those are for the individual assessments. For group requirements, group assessments, again, as I mentioned, TBA muna siya because it will depend on how the faculty will decide on whether a certain activity or worksheet will be given as a individual requirement, as an individual requirement, or as a group requirement. Okay, so for the course outline, we have 11 modules for our laboratory subject. I uploaded the syllabus in the transition e-class and also in the official e-class. So I hope you already downloaded your personal copy of the syllabus so you can go over it because it's going to be your guide for the whole semester. It will guide you what topics will be covered for specific periods of the semester. I'll have an overview of each topic. So for the first topic, actually the first topic is intended for reagent preparation, but as we've talked, as the faculty discussed, we decided to put it on a different position. The first activity that we're going to cover for the lab session next week is going to be the topic on cell ultrastructure. Here, we're going to learn how to differentiate and compare cells from two basic domains of life: the cells of bacteria, cells of archaea, and cells of eukaryotic organisms. So you're going to learn how to differentiate them based on their ultrastructure. Also, of course, to reinforce that objective, we need to appreciate how we use the electron microscope. So, majority of our discussion on this topic will also include how the electron microscope is working, how do we interpret electron micrographs. No? Now, going back to cellular structure, also you guys will learn how you can, how to, or not really how, but you will learn the basic functions of eukaryotic and prokaryotic ultrastructures. Of course, in theory, we apply, in theory, we know or we read about the functions of these structures. But of course, in order to add another layer of appreciation, we must see them, diba? Or we must be able to visualize them. So for this activity, that's the goal, to help us or to put in us yung extra layer of appreciation in terms of cellular ultrastructure, morphology, and function with the use of the electron microscope. The next module is the original one that is intended as Module 2, where you are going to revisit your knowledge about stoichiometry, about preparation of solutions. No? So you will be learning or reviewing how to perform mathematical computations in order to prepare the ingredients or prepare the components of a specific solution. No? Also, at this module, we will learn how to ensure an accurate preparation of a given solution by ensuring that the subcomponents are computed correctly and are and such computations, such accurate computations, will be the basis of correct preparation. The third module, or rather the fourth module, is on the function of the cell membrane. So the first part of the course, which is going to deal with cell biology, okay, so one of the modules that will be contributory to your understanding or to your newfound knowledge on cell biology is this module on cell membrane function. So we're going to examine active transport okay, using a model system, which is the cells, the epidermal cells, the epidermal cells of the frog skin. No? So we're going to conduct the frog skin bag experiment, and that will help us realize the important elements of active transport as a mechanism of cellular transport. There are a few specific objectives here that we'll just cover for the module itself, or when we already include when we are already in the module itself. The next module is on isolation of some organelles, no? Organelles ng mga eukaryotic organisms. So we're going to isolate chloroplasts, mitochondria, and nucleus. We're going to learn how to isolate them. We're going to demonstrate how they are isolated in the laboratory setting. Usually in the face-to-face, what we do here, we use we use plant tissues to isolate all these organelles, because these organelles, they can be extracted from a homogenized solution, from a homogenized cell solution, using different densities, no. So that's the principle of this module. No? Now, from one sample source, we're going to apply different work streams in order to extract the different organelles. No? In order to extract chloroplasts, mitochondria, and nucleus. At the same time, we are going to emphasize the instrument or one of the important instruments in cell biology, no, which is the centrifuge. Very important, because if you want to further study subcellular structures, it's a requirement that you be able to isolate them, and it can only be done using a centrifuge, using a refrigerated centrifuge. The next module is actually related to the last module, in the sense that in the face-to-face setting, what we do is whatever we get or whatever we discard from the previous module, we are going to actually use, in our for this current one, or for this module presented on the slide. So on this module, we're going to extract plant pigments and characterize them. Since we know that plants are photosynthetic organisms, they have photosynthetic pigments that are useful for them to catch energy from the sun or to catch light, and then utilize that for subsequent processes of photosynthesis. So in this module, we're going to demonstrate how you can extract photosynthetic pigments from plants, and then how can we characterize such pigments using chromatography and using spectrophotometry. Okay. The next module is photosynthesis. So the Hill reaction is for this activity, we're going to demonstrate the Hill reaction. So by demonstrating the Hill reaction, we will soon learn the relationship between light, the photosynthetic pigments, and the electron transport inhibitors that we will experimentally use in the lab in order to see how they affect the whole setup. No? For this activity, an emphasis will be given on how you treat data. Okay, so kinetic data will be collected in this activity, and you're going to process them in order to come up with rates, and also to be able to understand the whole biological activity of photosynthesis using a different lens, using a mathematical lens. Okay, so I think we're nearing we're now on the last half of the topics for the laboratory. Another module the next module after the Hill reaction is on cell cycle. So here, the goal is for you to have a visual appreciation of the different stages of cell division, from prepared micrographs. Usually, we use plant tissues to observe cell cycle divisions. So I'm sure you're very much aware of cell cycle divisions since as early as your first year, you already covered this topic. So for our module for this topic, we're going to, aside from appreciating, magkakaroon tayo ng secondary activities which include quantifying the cells in the prepared micrographs, analyzing the data that you will collect using software and using visualization tools. Okay, so I think these are the last two topics na. Ayan. So the last two topics will be will actually cover we'll cover molecular biotechniques. So we're going to isolate and characterize DNA, okay, and we are going to perform polymerase chain reaction, or we're going to demonstrate how polymerase chain reaction is performed. So as part of molecular bio, I think I can say that or I can say with confidence that one of the biomolecules that are well studied in the discipline of molecular biology is the nucleic acid DNA. So it's important that you get acquainted with how DNA is extracted from different matrices. We can extract DNA from living tissues, from humans, cells or tissues of humans. We can extract DNA from plants. We can extract DNA from bacteria, from fluid of animal fluid like blood or semen or saliva. No, so essentially, DNA is actually extractable from different living components or different living matrices. That's the focus for our module. But a growing or not a growing, but you can also extract DNA from environmental samples, but that's another story. So for our module, we'll focus on extracting DNA from biotic components, from biotic organisms. I think we're going to follow or get DNA from bacteria for this module. So we will demonstrate how to extract DNA following a protocol from the manufacturer of a DNA extraction kit. And I think we can also introduce an alternative method, or on my part, I'll make sure that I will include a discussion on how DNA is extracted alternatively if you don't have extraction kits. Kasi mahal ang extraction kits. So it's possible that you can extract DNA using Boyle lysis method, that you don't have to use any kit to get the DNA. Then after extracting, an important component is also characterization. So we will characterize our isolated DNA in terms of its purity and quantity following spectrophotometry. So we're going to use, actually aside from spectrophotometry, we'll also characterize DNA based on its visual appearance using gel electrophoresis. So those are two techniques that are worth studying for this module. And for the last topic, we're going to cover polymerase chain reaction. So here in this module, we will learn the basic principle behind PCR and how it is carried out in the lab. Okay. Also, we'll provide some case analysis on how you can interpret a DNA test that made use of PCR as a method for data production. The last module, which is I did not present here anymore, is on scientific paper writing. So we'll choose one topic from the final term that we will assign to the whole class, and you're going to write a scientific report, a narrative report about it based on the data that will be assigned to you. So if you will remember, aside from experimentation or appreciating experimentation, we are also teaching, or we chose to teach this subject following a research approach as well. So for module 11, or for the last module, we will cover that component, yung research component. Okay. So by this point, at this point, I'm pretty sure that you have a few expectations. So, and since we're conducting this in a synchronous method, I cannot get your expectations right away. So I want you to feel free to let me know of your expectations through the self-introduction output that I placed in our official e-class. So I want you to answer them, or I want you to express what are the things that you expect from the conduct and the delivery of the subject. Now, the course policies and reference laboratory manual that we are going to use are the next discussion points. No, I'm not going to discuss all of these, but some ano na lang siguro, no? Some important reminders. Okay, so for enrollment in e-class, since we're following an automated system, I will not be able to enroll students in our SBE class anymore. So if you notice, you were automatically enrolled in CILP produce e-classes. Those e-classes that you are in right now, I did not create them. So it's our CILP who created those e-classes. And the method of enrollment now, or the process of enrollment, is automated. So you cannot ask me to actually enroll you. It will be the system that will do that. Kaya nga what I did is I have a transition, I dedicated that, yung mga ginawa ng CILP, as transition classes. Then if you're already enrolled in that subject, you just have to get that to submit or answer the transition survey, and then you will be officially enrolled doon sa e-class na ginawa ko, where all our content, all our outputs, and our consultation will be done. So I don't have the capacity, again, to simplify everything I've said, I don't have the capacity to enroll you guys in the e-class, in the e-class provided by the CILP. You will be, though it's system generated, so we just wait for the system to include your name in the official class list. Okay? And you yourselves also, you will not be able to enroll your classmates, or I think even with the access code, wala, hindi siya mag-work. Okay? So I suggest you don't waste time in trying to enroll yourself kung hindi pa kayo naka-enroll via the system. So we have to wait for that. Now for communication and feedback, we communicate via Schoolbook, via Messenger, Facebook Messenger, and via MS Teams. Okay? So all your official questions, clarifications, you have to send via Schoolbook, via Facebook Messenger, or via MS Teams. And I will try to get back to you within 48 hours. Now, for the attendance policy, yun nga, I've discussed this in the class participation part. We will include attendance as component of the class participation. Okay? Then for intellectual property, an important component or policy here, whenever you submit outputs, you as much as possible try that you create your own diagrams, your own tables, your own discussions. Okay, because of course we shouldn't be tolerating copy pasting from original material of different individuals, because that's an intellectual property issue. So you have to, as much as possible, or if not, kung di naman kaya gumawa ng original content, then you can make sure that you do proper citation. Other policies will be attire, microphone use, video. So on my end, since we are including attendance sa ating class participation, for the first 5 minutes of our sync session, I will ask everyone to open their video so I can validate that you are really the one taking your classes. Then after a while, you can turn it off if you want to, or you can stay online if you're good with that. But on my end, I will require you for the first 3 or 5 minutes just for me to validate your attendance. Now, what else? Medium of instruction. Our medium of instruction is English. Okay, so my pre-laboratory sessions and post-laboratory sessions, all recorded sessions, I will be delivering in English. I will be code switching from time to time, pero it's going to be very minimal because I understand that some of you may be foreigners who are not familiar with the Tagalog or the Filipino, with Tagalog or Filipino. So we were doing the instruction in English. Now, the reference laboratory activities are lifted from the material of Dr. Tabo, Mr. Pareja, and Miss Tabo. So they have a standing cell and molecular bio lab manual that we have been using for a long while, for a long time already. So the outline of topics is actually lifted from the laboratory manual of these three faculty members. So all the things that I've mentioned a while ago, they are designed originally by Mr. Dr. Tabo, Mr. Pareja, and Miss Tabo. Okay. Now, personal rules, reminders, regulations that I will be imposing. Okay. So since I'm your instructor, there are things that I will be observing religiously every time that we conduct classes or every time that I interface with you or interact with you as my students. I start the class on time. Okay, so since we have 10:30 to 1:30, I will have my pre-laboratory discussion at exactly 10:30 a.m. Or sometimes 10:45, 10:35, some minutes to be provided, especially for students who are yet to what do you call this, who are yet to join. So I'm going to give extra minutes for consideration. But I start the class on time. Okay. Now, participation to our class discussions is important for you to have an authentic learning experience. So I know that it can be tiring to be energetic, to try to even be participative, and I understand that. But a tip from me is if you want to really learn the module, no, you have to be able to engage, or you have to be able to engage yourself to the discussion. So I will try my best to provide pre-lab discussions that are as engaging as possible, so I can catch your attention, and you will stay focused for probably 45 minutes of the pre-lab, and you'll, as much as possible, you can get substantial learnings from the pre-lab before you do the demonstrations, or before you carry out the virtual laboratory. I suggest that you should be prepared every time that we conduct our synchronous sessions. You have to read the material that will be provided to you prior to the class sessions. Third rule is bullying. I do not tolerate any act of bullying in my class. So proper decorum is expected from everyone. Okay. So on your end, since we're not, or on our context, since we're not on a face-to-face setting, it will be hard for me to actually sense undetectable bullying. So on my part, what I can offer is, if any of you feels na he or she is being bullied by others, he or she is not being treated differently by the group or by the class, feel free to let me know so I can try my best to help you. Okay? Kasi we're not in the face-to-face setting, so I have no way of telling whether one is already being bullied or not. Okay? Last reminder or regulation I want to impose. Academic dishonesty will not be tolerated, will be given special attention. And cases of verified or validated academic dishonesty will be directly endorsed to the Student Information Office. So now in our setup, the idea of academic dishonesty is something that is still hard to grasp. Yung definition natin ng academic dishonesty is continuously evolving given the distance learning setup. Like right now, I will have no way of really telling whether someone cheated or not, no, based on our current frameworks and current methods of course delivery. So on my end, if I ever encounter a validated academic dishonesty case, that's something that I don't tolerate. So definitely I will endorse that to the student information office for proper action to be delivered, no, for proper response to be given. Now, for the exams, this is how I will be providing exams to you. The enabling and summative assessments, they are time-restrained exams. And they can be a combination of questions such as identification, true or false, modified true or false. You enumerate, you analyze a problem or a case, or you solve a specific question or a specific problem or case. You provide computations, or you have to do computations in order to arrive to a solution or a parameter that is being asked. So this is how I will be giving the enabling and summative assessments. They are all checkable via the SB. So they are objective questions. Though may ano tayo, no? I can provide hybrid questions then, like essay within the summative or enabling assessments. And you just have to answer in free form. Okay. Consultation hours, I don't have consultation hours as of the moment. We haven't finalized our faculty loading forms, so we don't have consultation hours yet. However, you can contact me anytime, anytime. Okay? I'm online, or I can be contacted 24/7 via phone, via email, or social media, or via school book. But please, guys, do not expect an immediate response. So if I'm not online, then probably I won't be able to respond to you in real time. I will try to get back to your queries, to your messages within 24 or 48 hours. Okay? Usually naman, I don't keep messages unread. So as much as possible, I'll be able to, or I'll try to really reply to your concerns in the fastest way that I can. Now, I think sa Messenger, no, if you're not that we are not directly connected, some of your messages may be directed to the spam or the archive folder. So if that will be the case, make sure that you inform me via the chat, via our group chat, that you have a personal message to me, so I can so I can read and attend to your concern. Okay. So how do you contact me? It's here. My work email is shown on the slide, and also my personal emails if you have other questions that you want to course through on a different occasion or a different context. And you can also contact me via phone if an urgent need arises. Okay. Now for the references and the reading list. So here are the references that are going to be used for the entire semester. If there are additional references that I will use, I will make them available to you. Okay, so I think that's the end of our course orientation. So for the second part of, or the second meeting for the week, I hope that you guys will be able to go over the netiquettes and the tips for in order to succeed distance learning. I have already posted the content in our module number 1. Okay, that's it for this dedicated session. If you have questions about our course orientation, please let me know via our group chat or via the messenger or the school book platform.